Welcome back to all the students in this lecture 2 of plant pathology. Last time we had just understood just a brief introduction of what is plant pathology and what are its classification criteria, right? So today we are going to learn about general pattern of disease cycle, right? So disease cycle actually represents pathogen biology in the form of series of interconnected stages. These stages, they give an insight of how a pathogen sequentially and slowly is actually going to infect a host plant. So disease cycle is basically interconnected stages. These stages are inoculation, penetration, infection, growth and reproduction, and dissemination of pathogen, right? So these are series of interconnected stages, and a pathogen progresses from this one stage to second stage, and it slowly and sequentially it enters and invades a host plant, right? So we are going to understand each of these stages one after the another, right? So we start with our first stage of disease cycle, which is inoculation. Inoculation comes from the term inoculate, which means to introduce something, right? So inoculation word we also use when some vaccine is being used. Some vaccine, when it is introduced into human body, that is also called inoculation. Similarly, inoculation word is also used when we work in laboratory, and in that laboratory sometimes we use fungal and bacterial cells. We introduce it into Petri plates, so that is also called inoculation, right? So here inoculation here means arrival of pathogen on the host, right? So inoculation is the step when whatever the pathogen is, may it be fungi or may it be bacteria or virus or any other pathogen, when it arrives and sits on a host, it is called inoculation. So this is the first stage. Now an important term to understand here is inoculum, right? So what is inoculum? It is the pathogen that lands on the host, or we can say that it is a small amount of material containing pathogen, or we can also say that it is the active material, right? So inoculum is pathogen itself that has arrived on the host. When we talk about laboratory, then it is the active material or the some parts of pathogen, for example some cells of pathogen that are active, and we inoculate into Petri plate. So it is just a small amount of material that contains pathogen. So possibly it could be spores, right? It could be mycelium, hyphae of fungi, it could be bacterial cells. So this is called inoculum. It should be an active material, right? So inoculum is actually pathogen which arrives on the host, and this process is called inoculation, right? Now this inoculum can be of two types. This is primary inoculum and secondary inoculum. So primary inoculum undergoes a dormant period, that is, any primary inoculum will actually undergo a period of dormancy, a period of rest, right? After that period it will cause infection, so it is called primary inoculum. So any inoculum that is going to go through a dormant period and then it is going to cause infection to the new crop that has arrived, it is called primary inoculum. So primary inoculum leads to infection, that is called primary infection. So in other words, we can say that infection caused by primary inoculum is called primary infection, right? Similarly, secondary inoculum is the one that is produced after primary infection, that is, secondary inoculum is a result of primary inoculum, right? So whatever the primary inoculum has produced after infection will be called as the secondary inoculum. Now the infection that will be caused because of secondary inoculum will be termed as secondary infection. We will explain this term later on in the lecture more clearly, but for right now you can just keep in mind that primary inoculum is the one that goes through dormant period and that leads to primary infection, and secondary inoculum is the one that is produced from primary infection and this leads to secondary infection, right? So here a figure has been shown which shows that these all are fungal spores. Fungal spores are basically very minute, in very micron size it goes, and therefore it almost looks like dust. So these particles that are moving are actually fungal spores, and this is inoculum, right? So this is called inoculum, that is, it contains spores of fungi. So this is an active material, this is going to infect another plant, right? So this is inoculum. Now this inoculum could be primary and secondary, right? So now this inoculum, that is this spores or any other part of a pathogen, can be derived from various sources. This could be from the residues left in the soil. For example, when field is being cleared after farming, so there may be some residues left in the soil, some residues of the plant, and that plant might be carrying the inoculum. Then it might already be present in the soil. For example, all the crops has been cleared but the soil is there and some of the spores are present in the soil. Similarly, it could be present in the weeds or crops itself, in some of the debris of the crops, or in the weeds, whatever unwanted plant is present, the spores might be present on the weeds, that is unwanted plants also. It could get attached to farming equipment tools and that could be a source of inoculum. It could also infect seeds of plant. So the seeds, when you grow next time, that seed would already be infected, and when that seed is grown, the plant would be a diseased plant. Similarly, another source could be wind or water. Like wind and water, they can carry spores or inoculum of pathogen from one place to another. And also it could be carried by insects or animals or birds. When they sit on a flower or when they sit on a plant, the spores might get attached to their bodies, and this could lead to the spread of or the source of inoculum, right? So this is about inoculation, where a pathogen arrives and sits on a host, right? Now we move towards our next stage of disease cycle, that is penetration, right? Penetration, as the name suggests, it is penetrating or invading something, that is when something gets inside or it invades into something, it is called penetration. So here the pathogen which has arrived on the surface of the host plant will now try to enter the host. So this is called penetration, right? This is actually initial invasion, that is, the plant has actually not invaded, but the plant is trying to invade, so it is called initial invasion, right? Now this penetration can be of three types, can be made possible by three types. This is direct penetration, penetration by natural opening, and penetration by wounds, right? So let us first understand that what is penetration by natural openings. Natural openings are the openings that are already present in the plant. So these openings are stoma, right? Stoma or stomata, as we know, has guard cells, and this is used by plant for exchange of gases, right? So this is stomata. By this opening, the whatever pathogen is there, it can enter by this opening. Second is lenticel. These lenticels are raised pores, as you can see in the figure. These are present in woody plant, and it allows for gaseous exchange between the atmosphere and the internal tissues, right? So lenticel is also an opening. Similarly, hydethodes is also an opening. Hydethodes, if you know, is a modified pore on a leaf that exudes drop of water. So these are hydethodes. These are water droplets which are coming out from hydethodes, which is a pore, right? So these are usually present on the leaf margin. Sometimes we can see this kind of droplets coming out from leaf margin, and this actually comes from hydethodes. So hydethodes are also opening from plant. So these are the natural openings present in the plant, and the pathogen can actually enter from these natural opening, and therefore this kind of penetration is called penetration by natural opening, right? Now that we know what is penetration by natural opening, we move towards understanding of what is direct penetration. Direct penetration is where there are some specialized structures produced by fungi. These structures help in penetration, right? Now here no natural openings of the plant are involved. The fungi is capable of entering in the plant even without the natural opening, and therefore it is called direct penetration because it utilizes its own specialized structures to penetrate into the host plant. So let us understand this with a diagram. This single diagram will explain you all of the specialized structure that fungi produces to have direct penetration. So one such structure is apressorium. So first of all, let us know that this is a spore of a fungi. The spore, it lands over the epidermis of the plant. This is the plant epidermis, that is this is the leaf, and the structure of the leaf, that is the epidermis, the upper part, right? Now a spore has just arrived over the leaf, and the spore is germinating, that is the spore is producing germination tube, right? So after production of the germination tube, this bulbous structure that you can see is apressorium, right? Apressorium is a specialized cell which is used to infect host plant. This apressorium is a bulbous structure. It works on the principle of turgor pressure. As the pressure gets filled, it will give out penetration peg. Now this slender structure, thin structure that you see that arises from apressorium is penetration peg or penetration hyphae. This is leg-like structure. It gets inside the host cell, right? So this is penetration peg. It is also used to infect the host plant. So as I mentioned, the second is penetration peg, which arises from aprosorium, right? Then the next is intercellular mycelium. Now what is intercellular? When something is present between two cells, it is called intercellular. So this is mycelium. Mycelium is a vegetative structure of fungi. It is also called hypha. When it is single, it is called hypha. When it is many, then it is called mycelium, right? So when these hypha or mycelium, when it is present between two cells, it is called intercellular mycelium. Now this again infects the host plant. And then there is haustoria, right? So haustoria are these structures that has entered inside the host cell, inside the cell. So haustoria are for what? It is for absorption of water and nutrients from the host plant. So with the help of haustoria, the fungi will actually absorb all the nutrients of the plant, and that way it will keep on growing, right? So the next structure is haustoria. After that there is intercellular mycelium with haustoria. That means the intercellular mycelium that is present between two hosts produces haustoria. So haustoria or intercellular mycelium with haustoria are almost alike, right? So these are some of the specialized structure that a pathogen can produce for direct penetration. So from the spore, germination tube is formed that leads to formation of bulb like aprosorium. It under pressure produces penetration peg. This penetration peg further gives out intercellular mycelium, and this intercellular mycelium gives out haustoria, which absorbs nutrients from the host, right? So this is direct penetration. And the third type of penetration is by wounds. That is sometimes plant have certain kind of wound when we pluck a leaf, when certain abrasions are there because of wind, right? Because of water pressure, or sometimes when we pluck a flower. All these leads to some kind of injury, and through this injury, through this natural opening that has formed because of injury, the host can get infected, and this can lead to the penetration of pathogen into the host. So this is the second step or second stage, which is penetration. Okay, so now we move towards our third stage of disease cycle, which is infection, right? So infection is the establishment of pathogen in vulnerable cell or tissue, right? So what are vulnerable cells? These are cells that are exposed to the possibility of being attacked, right? So the pathogen which has entered in a cell, that cell would be a vulnerable cell because that cell has been attacked, right? Now since the pathogen has attacked that cell, so the pathogen will now try to establish itself completely in that cell, right? So that is infection. So why this establishment takes place? It is mainly for nutrients, right? So the pathogen will establish itself in the host in search of nutrients, because the fungi, the pathogen, it requires nutrients to grow, and that nutrients it will obtain from the host plant, right? So for the nutrients, it will start establishing itself, establishing itself into the host. In other words, it is going to infect the host, right? So infection occurs when when the pathogen successfully enters a host plant. So when it can be said that infection has occurred? When a pathogen has penetrated, successfully entered a host plant, then we can say that infection has occurred. So for infection to occur, the first and foremost requirement is penetration, right? So if penetration has occurred successfully, the plant is going to get infected. Now this infection will now lead to the appearance of symptoms, right? Symptoms are the visible appearance that we can see over the plant. This could be in any color, and this could be in various forms. It could be over the fruit, over the leaf, over the stem. It could be warty appearance, it could be smooth appearance, but it could have various colors. So these are symptoms, and this happens when a pathogen has caused infection in the plant. So now whenever you see a plant with symptoms, right, you can now say that the plant, the host plant is infected by a pathogen, and the pathogen has reached to the stage of infection, because of which now you can see symptoms, right? Now there are two words that you need to understand. One is incubation period. Incubation period is the time interval between inoculation and appearance of symptoms. So what is inoculation? Inoculation is the first stage where the pathogen arrived over the host plant, and appearance of symptoms takes place when the plant is infected. So the time between these two things, these two stages, is called time interval. The time interval between these two stages is called incubation period, right? And the second word that you need to understand is latent infection. Latent infection is when the host is infected with pathogen. That is, the pathogen is already present inside the host, but there are no symptoms. But there are no symptoms by which you can identify that pathogen, right? So a live example, a present example is of coronavirus. This is just to give you an example. There are cases where people are infected with coronavirus, but there are no symptoms visible. So when there are no symptoms visible, that does not mean that they are not infected, but they are not showing symptoms, but they have the disease, right? So that is called latent infection, and same happens in case of plants also. The plant may be actually infected by the pathogen, but it may not show disease. Then it is called latent infection. That means the symptoms will appear later on, though the plant is infected, right? So these are the two important terms that we need to understand. Now we move towards the fourth stage, which is growth and reproduction. So now the pathogen that has infected the host plant will now grow and reproduce. So to grow, it needs to reproduce. So reproduction is the process by which it will increase its progenies. So it will either reproduce asexually or sexually or vegetatively, and by this way it will multiply its number, and because of which there would be an increase in their number. So as the infection stage has occurred, whatever the pathogen is, it will start increasing its number, so the infection will spread more and more. That will it will spread all over the plant, so the number keeps on increasing. So this is the stage of growth and reproduction. Now the last stage is dissemination of pathogen. So disseminate means the act of spreading rapidly and widely. That is, something that spreads rapidly that is it is spreading fast, and widely, that is it is spreading over larger areas, right? So disseminate word can also be used for other purposes, like for example dissemination of information. So an information sometimes spreads rapidly and widely. So same is here the meaning with pathogen, where the pathogen will disseminate, will spread itself rapidly and widely. So this is dissemination of pathogen. This can also be called as dispersal, that is spreading. So now this dissemination could be by air, by water, by insects, by seeds, and by humans, right? So by air there could be two ways. First is that sometimes when the spore is present in the air, it touches wet surfaces. So it might touch wet surfaces like walls or like plant leaf, right? And it will get trapped in the water droplet, and there it will then start infection. And another method is when rain falls, it is brought down by the raindrops. So spores are moving in the air, and when rain falls, the raindrops brings the spores down, right? Then by water, it can spread through rain or irrigation water, or by lakes. This can go from one place to another, and the spores might reach there. The other way is by insects, right? Insects or birds, right, they feed upon the plant, and when they feed, sometimes these things, spores, the inoculum, they attach to the plant, and these insects, they move from one place, one plant to another plant, and that way it can disseminate the pathogen. And second and last is by seeds. So sometimes the seeds, the plant seed itself might be infected, and these seeds are sometimes transported from one field to other. So when these are planted, the new plant that will be formed will be infected. Similarly by human, so while handling diseased plants, tools or equipments, right, in the farm, so human can also disseminate the pathogen. So these are the ways by which pathogen can be disseminated. Now we just take a brief look at the whole process. So first stage is inoculation. We can also call it attachment, where the inoculum, it comes and sits over the host. Second is penetration. This is direct penetration. It could be penetration by natural opening or by wounds. After the pathogen has penetrated, now it will start the infection, and infection will lead to the appearance of symptoms. So after infection, it will reproduce more and more, due to which more number of spores will be formed, more number of hyphas will be formed, and this all spores that are produced will now get disseminated. So that means it will spread from one plant to another, or from one field to another. Some of these spores, they will go through the stage of survival, that is dormant period, where they will go into the period of rest. After rest, now this will again infect and go over a new plant, and it will cause infection. So like this, the disease cycle keeps on continuing. Now for your just understanding, here I am taking an example of loose smut of wheat, which is caused by ustilago, which is a fungus. Here I just want to make you understand how the disease cycle goes. So we start from this stage. This is a primary inoculum, that is, this has undergone a period of dormancy. This is a seed, right? This is an infected seed which was harvested. The infection is not visible. This is a primary inoculum. So this is without symptoms, but it is an infected seed. So this is a latent infection. So this seed, after harvesting, is planted in the next season, and this seed has been harvested because it did not show any symptoms. So the farmers thought that the seed has no infection. Now when the seed was planted, so the fungus now starts to spread into the plant, right? So this is the stage of infection, because penetration is already there in this seed, right? So now the whatever fungus is there, it will start penetrating and infecting the ear of the wheat plant, which is the main economic value, right? We get wheat from this ear. So it will start showing infection symptoms. So there is appearance of symptom. What is symptom? Powdery mass of spore is replaced by seeds. So all of the seeds are replaced by spores. That means the plant is infected and is showing symptoms, and also it is undergoing growth and reproduction, because more and more spores are produced. That means it is increasing its number. Now after this spores are produced, they will, by the wind, they will spread to other new flower, new plant, disseminate, right? So it will disseminate and reach a new plant, where it will infect the embryo and it will get infested into the seed. Now this seed will be harvested for the next season, and like this the infection will continue. So this is about how the disease cycle goes in loose smut of wheat. Now the next thing that I want to make you understand is a difference between primary and secondary inoculum. I am not going to make you understand a whole of this life cycle, because it is not important for you right now. What I want to focus on is primary inoculum. So these things that you are seeing, infested seed, diseased debris, right? These are primary inoculum, because these are going through a period of dormancy. Seeds, these are infected, but these are latent infection, not showing symptoms. Similarly, this is disease debris. Sometimes in the field, when everything is collected, some part of it remains in the field, so that remains as debris. This is diseased, so it would be infected. So this can this will remain dormant in the field until it gets a new host. So this will remain dormant until a fresh host is available to it, because right now there is nothing in the field to infect. Everything has been harvested. So as new plants will be planted, these will get inside the host plant, right? So this is primary infection, this is primary inoculum, and the infection that they have caused to the new plant is primary infection. Now because of primary infection, the plant will get diseased and it will produce other spores again. This is secondary inoculum, because these spores are produced by primary inoculum. After primary inoculum has infected the plant, the next stage of spores that are formed are called secondary inoculum, and the infection caused by them is called secondary infection. Now this secondary inoculum will cause infection and it will cause disease to plant, but some of it will again reach to the soil, it will reach to debris, and this will go into a period of dormancy until a fresh host is available. So this is primary inoculum. I hope this is clear with you. For now you don't have to understand this cycle, because you have already a separate syllabus of this is white rust of crucifer. You have a separate syllabus for it, and similarly you have a separate topic of loose smut of wheat, which I'll be explaining in later lectures. So for now this is just for you to understand that what is primary inoculum and what is secondary inoculum, right? So with this we come to the end of our session. So I hope that you all will clear your doubts regarding this. If you have any doubt, you can just contact me, and I'll be back with the continuation of this topic later on. So till then, take care. Thank you.